Greetings all, my name is Muhammad Yassin. Today I'm going to present about recent advances in messenger RNA vaccine technology, discussing mechanism, application, challenges, and future perspective for infectious disease prevention. The global burdens of infectious disease, there's no doubt that now infectious disease have a negative impact on public health and economy. However, vaccines remain one of the best strategies for preventing infectious disease and reducing morbidity and mortality. We have tuberculosis, malaria, and HIV. Those account for the majority of worldwide disability-adjusted life years. And low-income countries are mainly affected by illness that are otherwise preventable. Conventional vaccine platforms such as live attenuated, inactive, and etc. Prevent many diseases, but have many limitations with rapidly evolving microorganism, safety profile, development time, and genetic variability. On the other hand, the messenger RNA, which is first proposed in 1970 and demonstrated against influenza in 1993, messenger RNA vaccine have evolved into rapid, safe, effective platform. In addition to that, the COVID-19 was successfully enabled by lipid nanoparticle delivery, scalable manufacturing, and molecular engineering, establishing messenger RNA vaccine as promising strategy for preventing future infectious disease. This is the roadmap for my presentation. First, the structure of the messenger RNA vaccine. The messenger RNA vaccine have 5 structure elements. Those work together to enable stability, efficient protein synthesis, and we have the 5-cap, 5 and 3 untranslated region, open reading frame, and polytail. The traditional messenger RNA vaccine, we have single-strand RNA, which is consists of from 1,000 to 5,000 nucleotides, the same structure that we have discussed before. The production of this messenger RNA vaccine by in vitro transcriptions using linear plasmid DNA, PCR template, bacteriophage RNA polymerase, ribonucleotide, and RNase inhibitor, and purified by post transcriptions, which is removing the residual DNA by the enzyme DNase. Compact forward structure, desirable for production and distribution. However, this technique have limitations such as short half-life and transit protein production require large doses. On the other hand, the self-amplifying RNA, which is consists of 9,000 to 12,000 nucleotides. We have alpha virus, which considered the backbone, and also replication complex. The replicase protein translates first, producing subgenomic and negative sense RNA intermediates that amplify target protein expression. One of the advantage of self-amplifying RNA, messenger RNA, is need only low dose and you get high output. And we have trans amplifying system, which split replicase and gene of interest into separate transcription to overcome delivery challenge for large genome size. This is the delivery system. Um, as we know that the lipid nanoparticle only clinical authorized delivery method for RNA therapies that proven in vivo for medication, biomedical, biomolecules, and vaccines. We have three types, which is polyethylene glycol lipids, cholesterol, and cationic lipids. The delivery system, this is the core, as we discussed before. Messenger RNA vaccine manufacture process. We have two type, upstream and downstream. The in vitro, the upstream production in vitro transcription using recombinant plasmid with the target gene, RNA polymerase, nucleotide, magnesium buffer, RNase inhibitor, plus polymerase, and methyl transferase, guanine transferase, pyrophosphatase, and etc. And the downstream purification, eliminate contaminants, enzymes, plasmid DNA, double-strand RNA, residual nucleotide, and truncated transcription. And we have reverse phase HPLC had actually boost translation efficacy and protein expression by from 10 to 1,000 fold. This is the immunological response to messenger RNA vaccine. After injected the messenger RNA vaccine, lipid nanoparticle encapsulated messenger RNA is taken by the muscle and tissue resident antigen present cell, particularly dendritic cell, via endocytosis. Once it's released into cytoplasm, the 5-cap untranslated region and polytail promotes RNA stability and efficient translation by host ribosome producing encoded antigen. The antigen then stimulates both humoral and cellular immunity responses. In the extracellular or release antigen is taken by antigen present cell ABCs and present on the major histocompatibility class 2 molecules, which is activate CD4 T helper cell, and then supported by B cell activation and antibody production. Intracellular synthesis of antigen is present by major histocompatibility class 1 molecules, and dendritic cell also cross present antigen, thereby activating CD8 cytotoxic cell and eliminate infectious or antigen express cell. In addition to that, we have germinal center response for generating B memorial cell and long plasma cell for durable immunity. This is the process as I discussed before. Now this is the clinical application for messenger RNA vaccine. We have in the COVID-19, which we everyone I guess had these shots. You receive maybe Moderna and Pfizer BioNTech. We have bivalent, trivalent, pentavalent, and octavalent COVID vaccine for dengue fever, also Ebola, HIV is still on clinical trials, tuberculosis also, and influenza. Advantage of the messenger RNA vaccine include the safety, since it's not integrated into DNA, host DNA, and are non-infectious also. The efficacy or effectiveness, nucleotide modification improve immunogenicity and translation efficacy. Manufacturing, we have the cell-free produce is rapid, scalable, and cost-effective. In addition to that, the ability of messenger RNA can encode multiple antigen simultaneously, boosting the immunity against resistant diseases. However, this messenger RNA vaccine have some limitations, such as autoimmune disease or autoimmune reactions such as myocarditis and autoimmune thrombocytopenia. In addition, polyethylene glycol can cause hypersensitivity for some patients. And one of the major limitation of messenger RNA vaccine is called chain, which this vaccine need ultra cold chain actually. Advanced purification and AI-assisted lipid nanoparticle design. The purification and quality control, we have these contaminants such as double-strand RNA, enzymes, and residual DNA templates are common in vivo transcriptions. This can reduce stability and trigger adverse immunity response. As we mentioned before, that the reverse phase HPLC is particularly effective, increase messenger RNA translation up to 10,000 folds. The AI and computer aided design accelerate the discovery for ionizable lipids, enabling rapid exploration of topologies that maximize delivery and stability. Machine learning models also, such as artificial neural network and light GBM, have identified lipid feature linked to improve delivery efficacy, and targeted delivery to specific tissue remain one of the challenge for messenger RNA vaccines. To overcome storage degradation and number of doses, there's many actually examples, such as lympholyzed, which is done by Moderna for cytomegalovirus vaccine. This technique actually increase the shelf life of the vaccine in 5 degree up to 15 months, and from more than actually 8 months in room temperature, and 21 months at 4 centigrade. The circular RNA also a new technique that which is covalent closed structure, give circular RNA greater resistance to endonuclease degradation and extend protein expression compared with linear messenger RNA vaccine. And self-amplifying RNA, as we discussed before, which is actually give high output with low doses. To conclude, advance in messenger RNA vaccine engineering, lipid nanoparticle delivery, and large-scale production enable vaccine that elicit robust immunity with a favorable safety profile, and also success against COVID-19 has intensified more research into other diseases such as influenza, TB, dengue, Ebola, and HIV. Remaining obstacle, thermal stability, ultra cold storage dependency, limit tissue distribution, production cost, and rare immunological adverse events. Also, AI assist lipid design, self-amplifying RNA, circular RNA vaccine, and lympholyzed formulation offer promising route to greater stability, lower storage, and wider global access. Continue multidisciplinary research in molecular biology, immunology, nanotechnology, computational modeling, and pharmaceutical science will shape future pandemic preparedness. Thank you for watching, and if you have any question, don't hesitate to send to my email. and this is the reference.